Good morning, class. Today in this class, let us go through important questions. So for whatever the units completed under fifth semester for the academic year 2023 and 2024. Okay, these are questions only for your particular test. Let us go through now 2 marks questions. First question, DNA ligase. You need to write what is this DNA ligase and their functions. Okay, 1 or 2 applications. Then phosphatases, cloning vectors, polymerases, CRISPR-Cas9. This is 2 marks, so kindly go through only definition and example. Like that, that will be more than enough. Then scope of genetic engineering, MCS, that is multiple cloning sites. This MCS, which comes in your PUC vector. Okay, please go through that one. Who discovered PCR? Give their types. Who discovered or who proposed this PCR? Then any 2 types, that is more than enough. Then you need to define annealing. This annealing which comes in PCR again. Stacked DNA polymerase, again this comes in your PCR. You need to define what is the stacked DNA polymerase and which organism this stacked DNA polymerase was isolated. Then what are the different methods of gene delivery? You have this in your syllabus. You need to just mention what are the different types of gene delivery, like physical method, chemical method, and about electroporation, microinjection, like that. Okay, kindly refer your syllabus copy. Then expand YAC and back. This full form you need to write. YAC carries 1 mark, back carries 1 mark. Mention the applications of cloning vector in genetic engineering. Any 3 to 4 applications and for your 2 marks. Then scope of genetic engineering, you need to mention here. What are phage and cosmids? Just need to define how phage we are using as a cloning vector. Cosmids also one of the cloning vector. Phage is also one of the cloning vector. So I need to define what is this phage and cosmids in genetic engineering. Then what are molecular probes? You are going to get this molecular probes in blotting techniques. Just define what are molecular probes. Okay. Probes. List different blotting techniques. Just need to list the different blotting techniques like southern blotting, northern blotting, western blotting. You need to mention. Then what is gene knockout techniques in bacteria? Just define what is gene knockout in bacteria. That is enough. So these are some of the important questions for your first internal test. You can go through. Next question for 5 marks. In that first question, write short notes on restriction enzymes. What are PUC vectors? Explain with suitable example. Any one PUC vector, you can draw the diagram and explain. Write short note on electroporation and micro injection. Use of genetic engineering in development of therapeutics and vaccines. Next, write a note on enzymes used in recombinant DNA technology. So whatever enzymes which comes in genetic engineering, which was explained in class now, all the enzymes, you need to write it here. Mention importance and applications of genetic engineering in various fields. Here, in general, all the applications you need to give here. Then 7th one, discuss industrial applications of genetic engineering. Here, the industrial point of view, what are the applications of genetic engineering that you need to mention. Write a note on southern blotting techniques. Okay, so here, what purpose we are using southern blotting, how the southern blotting will be done, that you need to focus. You will get notes, go through the notes also. Explain the construction of recombinant DNA technology. So all these questions which comes under 8 marks. So keep in your mind, in your internal test or maybe university test paper, you will get 2 marks, 5 marks and 8 marks questions. So be prepared according to that particular marks. Explain PCR and its applications. Here you need to explain the PCR 3 techniques, then give their applications. So third one is what are plasmids? Give the structure and characteristics of PBR322. Explain industrial applications of genetic engineering. Then what are the different types of blotting or techniques? Okay, then explain any one method of blotting techniques. So these are your important questions for your first internal test. 2 marks questions, you can take a screenshot. Then 5 marks questions. and 8 marks questions. I hope today, whatever this slide have made regarding your paper 5 important questions will be really helpful to score very good marks. Thank you everyone for being with me.